How many were here last night? Okay, how many were not here? Okay, that helps me a little bit because there's a few things that we can briefly review without belaboring. Okay, when Darwin finished his famous masterpiece, The Origin of Species, he thought he had explained every clue but one. And the clue that he knew he hadn't explained had to do with an extraordinary event in the history of life known as the Cambrian explosion. Now, the Cambrian explosion is the geologically sudden appearance of most of the major animal groups or body plans, the body plans that are exemplified in the animal groups. A body plan is a unique arrangement of body parts and tissues. And during the Cambrian period, a vast number of new animals, each exemplifying a new and unique way of arranging body plans or body parts and tissues, arose, unique body plans. For example, and this was an animal that Darwin knew well, the trilobite, which is this guy on my far left, your far right, has an external skeleton. It's an arthropod, and all arthropods have articulated external skeletons. The trilobite has compound eyes, it has a gut. There's a whole interesting architecture to this animal that's unique and different from other types of animals with different body plans. The animal just next to it is a primitive fish, which is a chordate. And instead of an external skeleton, chordates have an internal notochord or spinal column. And so you can see that if you put two different animals together along those two different plans, there's going to be a lot of differences. And so during the Cambrian period, we have the first major groups of animals exemplifying these different ways, these different architectures, these different ways of putting together animals arising in the fossil record. Now, Darwin knew about this principally from the trilobites and another creature called the brachiopod, which is over there, which were well preserved in the fossil beds in Wales, in a place in Cambria, Wales, and that's the root of the word Cambrian explosion. And so I love this picture because this really conveys in one illustration the idea of the Cambrian explosion. We have the geological column on my left, and if you remember your basic geology, the sedimentary rocks stack up over time. All other things being equal, the oldest ones tend to be on the bottom and the younger ones tend to be on the top. And so at a certain layer, at a particular layer, and we now know all around the world, there are, corresponding to this Cambrian period in geologic history, we find the first major animal groups arising in the Cambrian fossil record. So if you think of this in terms of Earth history, the Cambrian is a period within the Paleozoic, and that is typically dated the Cambrian period is typically dated between 543 million and 490 million years ago, and the Cambrian explosion takes place within a narrow window of about 10 million years, with the most explosive period of the explosion in about a 5 to 6 million year window. Now that sounds like a lot of time, geologically speaking, but or it's a lot of time in relation to our lifespan or to even human history. But geologically speaking, that's an explosion. That's a very brief window of time. And we're going to see also that biologically speaking, that's a very small window of time because it's possible to quantify how much evolutionary change should take place if we know some key factors like the mutation rate, the generation time, the population size of various organisms. And knowing those factors, biologists can calculate how much evolutionary change should take place in a given period of time. Turns out that 5 to 6 million years, or 10 million years, or even 40 to 50 million years is not nearly enough time to generate, by the standard Darwinian mechanism of mutation and natural selection, all the intricate structures and anatomical features that arise with these Cambrian animals. So this is The Cambrian explosion is an explosion geologically speaking because the appearance of the fossils are very abrupt in the fossil record, but it's also an explosion biologically speaking because this happens much more quickly than the natural selection mutation mechanism could conceivably produce the amount of new anatomical structure and novelty that arises. So here's a picture of what Darwin what troubled him. The gold little dots on the top represent the new forms of life that arise in the fossil record. Darwin thought that the new forms of life, new forms of complex animal life should arise very gradually. His picture of the history of life, as we talked about last night, was this great branching tree, where the base of the tree represents the last common ancestor of all life, the first organism, probably a simple one-celled creature, and then all the connecting branches represent those forms that have gradually evolved, morphed, and changed from that first simple form. The first animals should therefore also should be represented. The history of the first animals should also be represented by a tree. And what we see in the fossil record instead is the first animals arising, but the ancestral precursors and intermediates between those forms of life are absent in the lower Precambrian strata. And this was a pattern that Darwin knew very well, and it troubled him. So here's what this picture depicts what you would expect to find, the tree-like picture. The gold dots represent the data, the blue dots represent those forms of life that we expect, but which we have not found, or which paleontologists have not found. So if we look at the raw fossil data, we have a picture of not so much of a tree, but something much more like a lawn, or perhaps an orchard of separate trees where there's very small variation within limits, but separate organisms arising very abruptly in the geological column. So the theory on the bottom, the Darwinian theory on the bottom, does not match the evidence or the data on the top. And this is the essence of Darwin's doubt. And so, as he put it in The Origin of Species, the question as to why we do not find rich fossiliferous deposits belonging to these assumed earliest periods prior to the Cambrian system, the system of geological layers, he says, I can find no satisfactory answer. And he went on to say that the Cambrian explosion, the explosion of animal life, was a valid objection to the views here entertained. This was in The Origin of Species. All right? Now, very few. Now, in contrast to the rhetorical modesty that Darwin expressed, Darwin was obviously convinced that his theory was correct, but he was enough of a scientist to acknowledge the difficulties, the anomalies, the questions, and his own doubts. But in contrast to that rhetorical modesty, today's modern spokesmen and women for Darwinian theory are increasingly known for their dogmatism. And I had a particular experience of that myself when I was here in the great state of Texas in 2009. I was testifying in front of your State Board of Education about the education policy standards that the board was adopting. They were considering adopting as part of their TEKS, their Texas Educational Knowledge and something, skills, thank you very much. They were considering adopting a proposal that would encourage teachers to teach the strengths and weaknesses of competing scientific theories. Sounds pretty non-controversial, uncontentious. But the Darwin-only science lobby turned out in force to oppose these standards. And their argument was, believe it or not, and this was clearly stated by Eugenie Scott, the head of the National Center for Science Education, as quoted in the Dallas Morning News, there are no weaknesses in the theory of evolution. You cannot apply a strengths and weaknesses pedagogy to evolutionary theory because evolutionary theory has no weaknesses. It explains all the evidence. Now, one of the themes of my new book, Darwin's Doubt, is that there is a huge disparity, and I mentioned this last night in the question and answer time. There is a huge disparity between the public presentation of the status of contemporary Darwinian theory by people like Eugenie Scott and Richard Dawkins and Bill Nye the Science Guy and the actual status of the theory as you find it in the peer-reviewed scientific literature. In other words, I call them Darwin's public defenders. And Darwin's public defenders are extremely dogmatic. Bill Nye is one of Darwin's public defenders. They're extremely dogmatic. They tell you that there are no problems, no weaknesses, everything is all sewn up, and yet when you get into the scientific literature, you find that there are very grave problems that are being openly acknowledged by leading evolutionary theorists and biologists, some of whom are now calling for a new theory of evolution because they recognize that natural selection and random mutation does not have the creative power necessary to produce things like whole new animal body plans and organs and structures. So there's this disparity between the public presentation and the actual status of the theory. Another of Darwin's public defenders, who is also known for his rhetorical excess, is Richard Dawkins. He said in the New York Times a few years back, "It is absolutely safe to say that if you meet somebody who claims not to believe in evolution, that person is ignorant, stupid, or insane." And then he very graciously says, "or wicked," but I'd rather not consider that. So that was nice of him not to consider the wicked option. Now, I wonder if he would apply this to, for example, James Shapiro, the leading molecular and cell biologist at the University of Chicago, who is developing a new theory of evolution because he thinks neo-Darwinism is so obviously inadequate. This is the kind of disparity that's taking place. In any case, Darwin, as I mentioned, was open about the problem posed to his theory by the Cambrian explosion. And it was a problem for two reasons. One, because it didn't match the tree-like picture of the history of life that he envisioned, and secondly, because his mechanism of natural selection required a vast amount of time to work. And he understood that even in the 19th century. And so the suddenness or the abruptness of these new fossil forms in the sedimentary record was a challenge to both parts of his theory, to the mechanism of natural selection and to his theory of universal common descent, which he represented with the branching tree picture of the history of life. Now, Darwin was, of course, enough of a scientist to, first of all, to address this mystery head on, to acknowledge that this was a mystery. And I call this first mystery the mystery of the missing fossils. But he was also enough of a scientist to come up with at least an idea of how the problem for his theory might be solved. And the problem, or the auxiliary hypothesis that he came up with, this supplementary hypothesis, it now goes by the name of the artifact hypothesis. And the idea of the artifact hypothesis is that we're not finding those missing fossils either because they are they were too soft or too small to have been preserved, or because we haven't looked hard enough. The missing fossils are missing. The reason the missing fossils are missing is an artifact of either incomplete sampling of the fossil record or incomplete preservation. And Darwin himself sketched this out in a very rudimentary form. He said in The Origin, he said, "I look at the natural geological record as a history of the world imperfectly kept. On this view, the difficulties discussed above, the difficulty of the Cambrian explosion, is greatly diminished." Okay? The missing fossils, the problem of missing fossils, can be explained away by assuming that either the fossils have not been preserved completely or we haven't found them all because we haven't looked hard enough. Okay? So that was how he thought the problem might be solved, and he looked forward to future fossil discoveries to vindicate his overall picture of the history of life. As I mentioned, he knew about trilobites and brachiopods and a few other main forms in the Cambrian. So what has future fossil discovery shown? Has it resolved the mystery of the missing fossils, or has it made the problem more acute? In fact, it's the latter. It has made the problem greatly more acute. And there have been two major finds that have emphasized or exemplified this point. The first is the discovery of the Cambrian fossils in the Burgess Shale in western Canada, in a place near Field, BC, British Columbia, just across the Albertan border. These were discovered in 1909, first discovered in 1909 by Charles Walcott, a now famous paleontologist at the Smithsonian Institution in Washington, D.C. He was prospecting in the Canadian Rockies when he found this famous gray shale called the Burgess Shale with these exquisitely preserved remains of Cambrian-era creatures. The creature behind me is known as, it's an arthropod, another type of arthropod. It's called Morella splendens, and it's a gorgeous little lace crab with beautifully preserved appendages. And I have an artist's rendering of this with white on black that really highlights the structure of this amazing creature. Another creature that was discovered, another arthropod, not all the forms were arthropods, but some of the best preserved were these arthropods with their hard exoskeletons. This is called a waptia. It's a kind of crustacean. You can see the little articulations in the tail section. It looks a little bit like a shrimp in that part of its body. And here's another, an artistic depiction of it with another fossil preservation. You can see the hard head shield and the beautiful segments in the body, all beautifully preserved. Now, these are just a few of the many forms of life that were discovered at the Burgess Shale. And I think I have a little model of another guy that, oh yeah. Some of them were extremely bizarre, strange anatomical, sorry, visual aids flying everywhere. This is a little model of a guy called Weewaxia. And you can see just, it's an animal. It moved along the bottom of the ocean. All these were marine invertebrates, these Cambrian-era creatures. And you can see he's just got some amazing structures, and you can kind of look at this. This is one of many examples. There's another guy I like called Opabinia that had 5 eyes and about 30 segments in his body. And Stephen Jay Gould made these fossils famous in a book called Wonderful Life because some of the creatures that were discovered were so exotic and completely unexpected in the uniqueness of their body plans, their architectures. So the Burgess Shale actually accentuated the problem of the Cambrian explosion. It made it more acute for 2 reasons. First, because it documented many more forms of Cambrian animal life than had been known in Darwin's time. The breadth of the explosion was much greater. There were a lot more new animals that arose abruptly to explain the origin of. And secondly, the time window of the explosion, as geologists were able to date the explosion, began to shrink. And so the explosion, in a sense, became more explosive. And this was, and has been, a real difficulty for paleontologists since then. The explosion became even more explosive, at least we learned that it was even more explosive, because of another extraordinary find that took place in southern China, it was first discovered in southern China in 1984. This is the famous Chengjiang locale where the Cambrian fossils are so exquisitely preserved, they look oftentimes like photographic images on the mudstone. There was a Time magazine article about the Chinese fossils in 1995, and it was titled "Evolution's Big Bang." And one of the paleontologists quoted in this article said, "What I like to ask my biologist friends is this: how much faster does this event have to be before we stop calling it evolution?" Because it was as a result of these Chinese fossils that the most explosive window within the Cambrian explosion was dated at between 5 and 6 million years. And this is, again, both geologically and biologically speaking, a blink of the eye. And so this is a very exciting discovery. And in the year 2000, we had the opportunity at the Discovery Institute to host a lecture by one of the leading Cambrian paleontologists from China. And we sponsored the lecture at the University of Washington, and it was really a very exciting and somewhat tense event. He came bearing many fossil samples, and so it was irresistible to the geologists, the paleontologists, the evolutionary biologists. They came to hear the lecture. What we knew was that this leading Cambrian paleontologist had also publicly expressed skepticism about Darwinian evolution in People's Daily, no less. Within the Communist Party official newspaper in China. So anyway, this Chinese professor gave his talk, and the first part of it was just electrifying as he showed the images, both fossil, and these are actually some pictures that we are using with his permission. And these were drawn by Chinese artists, but also with the fossil. So this is a guy named, this fossil is named Anomalocaris, and it was the largest predator in the Cambrian seas, and it got up to be about 1 meter long. So this is kind of a scary dude, and the appendages and the body of this guy were preserved exquisitely. But the different animals that were discovered, some of which with, again, completely unique body plans, made this Cambrian explosion, made our understanding of the Cambrian explosion that much more mysterious, because the breadth of the explosion was so wide. There was so much what paleontologists call morphological disparity. Completely different body plans, different animal structures. So here's a guy called Hyolith. He's got a conical shell with an attached lid. This is a filter-feeding worm, a phoronidid. You see both the artist's depiction and the fossil side by side in all these pictures. Another kind of worm, an annelid with a segmented body with lateral bristles for locomotion. This looks like a plant, but it's actually an animal that had these petal-like bracts in a feeding bowl. And the paleontologists were so perplexed by this, they weren't able to classify it. They just called it a problematica. And then this is another fossil. This is also from the formation over there is called the Maoshishan Shale near Chengjiang in southern China. I absolutely love this fossil. It's a comb jelly, and we have these in Puget Sound. And this is a great example of what paleontologists call stasis, which is prolonged stability in the form of an animal through geologic time. We have these today. I have a picture of a modern comb jelly with the, let's see, he's on my right, and then the fossil form behind me, and you can see that it's an identical structure. This is 530 million years of no directional evolutionary change. It's extraordinary. And then one of the great discoveries of the Chengjiang locale has been made more recently. The paleontologists have turned up a couple different types of primitive chordates, including one which is clearly a fish. And that's just incredible. The fish had previously been known as far back as the Devonian, I think, and now they have been established to have also come about in this explosion of animal form in the Cambrian. So this Chinese paleontologist is walking us through all these extraordinary discoveries. And at the end of his talk, he talked about the theoretical significance of the discoveries, and he put his hand like this, and he said, he said, the strange thing about these Cambrian fossil finds is that and then he did this, he said, is that they turned Darwin's tree upside down. Instead of having, the simple forms at the bottom gradually morphing and then the complex forms arising and branching out, he says we have the disparity between form at the very beginning of the explosion with nothing underneath. And so there was some uncomfortable shuffling, and then we went to the Q&A time. And one of the geologists from the University of Washington raised his hands, and he said, almost as if in warning, he said, "Professor, aren't you a little bit uneasy about expressing skepticism about Darwinian evolution coming as you do from such an authoritarian country?" And suddenly you could, cut the tension with a knife, to use the old metaphor. And but this Chinese professor, no one's fool, got a wry smile on his face, and he said, in our country, we can question Darwin, just not the government. And then he said, in your country, you can question the government, but you can't question Darwinism. And it was his way of saying, we've heard about political correctness in your universities. Whose country is more free, really? And I think some of the stories Bill Dembski was telling last night illustrate maybe this professor was all too well aware of. In any case, the Cambrian fossils have turned Darwin's tree upside down, and the mystery of the missing fossils has, in the ensuing 150 years, become much more acute. The explosion took place faster than Darwin knew, and the breadth of the explosion is much wider. Now, there have been a couple of different attempts to solve this problem. Here's an interesting quote. This is from Dawkins. And Bill mentions it, sometimes we really appreciate him because of his candor. He says, it's as though they, speaking of the Cambrian animals, were just planted there without any evolutionary history. Remember, we have to constantly keep in mind that these creatures were not designed, but rather evolved. Okay. Well, let's go back to the artifact hypothesis, because that has been the go-to explanation for why we don't find that evolutionary history. And the claim has been that the ancestors are seldom fossilized because they either lacked hard parts or because they were too small. And here the Chinese fossils have again made the mystery more acute, because in addition to the fossils found in the Cambrian layer, there have been some fossils found in the Precambrian. In a formation called the Dushantu shale, there have been some small microscopic sponge embryos discovered, likely sponge embryos. Sponges can be identified because of their distinctive spicules, and a Chinese-American paleobiologist, Paul Chen, who is actually a fellow of the Center for Science and Culture, has done work over in China with some of the Chinese paleontologists. He was a sponge expert. He did work in his PhD on this, and he was able to cross-section these microfossils, and he determined that they had these distinctive spicules of sponges. Either way, what's been discovered is small, soft-bodied organisms with small, microscopic, soft-bodied organisms. Wait a minute, stop, press, that's really important. Why? Remember, the artifact hypothesis said that depositional environments weren't capable of preserving the soft, small ancestors of the Cambrian animals. Well, wait a minute. If they can preserve these sponge embryos, then why can't they preserve all the other ancestors of the other sponges are primitive animals. They're very simple animals, and we have known for a long time that they first originated back in the late Precambrian. Okay, a little context. But they're not ancestral to trilobites or chordates, or this is not a plausible near ancestor of any of these other animals. So the point is that if you can preserve these sponge embryos, why can't you preserve the presumed ancestors of all the other animals that first arise in the Cambrian? The depositional environment is perfectly capable of preserving small and soft organisms, but for some reason did not preserve the ancestors to all the other animal forms that arise in the Cambrian period. And so many leading Cambrian paleontologists are now openly saying, look, we have to respect the fossil data and accept that the Cambrian explosion is a real event. It's not just an artifact of incomplete sampling or preservation. Two leading paleontologists who have a book out on the Cambrian explosion, it came out last year, same year as mine, Valentine and Irwin, have very openly argued for the Cambrian explosion as a real event. It cannot be explained away on the basis of being an artifact of bad sampling or preservation. So here's a nice little picture of this guy in cross-section. So that's the first mystery, and it's been made much more acute, the mystery of the missing fossils. In the book, and this is what I do in the book, and if you're intimidated because it's fat, don't be. Because what I try to do is I'm telling a scientific mystery story. And the first mystery is the mystery of the missing fossils, and it goes back to Darwin's time. And what I do in the book is I look at Darwin's doubt, and then I trace what has become of that mystery, and I show how it's become more acute with each progressive attempt that has been made to resolve it. And one of the reasons it's become more acute is that the mystery of the missing fossils is not the only mystery. The fossil explosion, the explosion of new animal life, bespeaks another mystery. And that mystery, I argue, is even more profound. And that is the mystery of the origin of genetic information. Essentially, it's an engineering problem. It's the problem of how you build an animal. And what we know, because of discoveries in the second half of the 20th century, is that building an animal, building a new animal body plan, building a compound eye, or any of the other intricate organs and structures and tissues that arise with these new animals requires instructions. It requires genetic information, and indeed other forms of information, which I'll talk about in a little bit. Key discovery, 1953, we talked about it a little last night. Watson and Crick elucidate the structure of the DNA molecule. 1957, Francis Crick, who was, by the way, a code breaker in World War II. Did you know that? His whole career is really fascinating to me. He was a genius. And he cracked, I think, the most important code, the genetic code. And he had what I think is perhaps the most important insight, hypothesis in modern biology, which is called the sequence hypothesis, when he proposed that the 4 chemicals that run along the interior of the DNA molecule, which chemists represent with the letters A, C, G, and T, are functioning like digital characters in a section of machine code. This is literally stored encoded information in a long chain-like molecule. Okay? So Bill Gates has said DNA is like a software program, only much more complex than any we've been able to devise. It's really like the encoded information that we use in high-tech digital computing. And what it does, and I talked about this last night, the information in DNA codes for the production of protein parts, enzymes or parts of molecular machines, and therefore this information is indispensable to building any kind of new animal life. All right, and I'll talk about that a little bit more in a minute, but I want to just make one key distinction. Engineers, scientists will know about what's something called Shannon information, or the information theory. And that's a mathematical representation of information that doesn't take into account whether a sequence of characters is meaningful or functional or not. Okay? So when we're talking about information in a biological sense, we're not talking about information in that purely mathematical sense. The top string of characters has information according to information theory, but it's not functional. It doesn't perform a communication function. The bottom string of characters does perform a function. It is specified as well as complex or improbable. And Bill Dembski talked about this string of characters would be very improbable. The odds of getting either one of those strings is going to be very small if you just sit at the typewriter and type with your people don't even use typewriters anymore. The keyboard, if you just type with your eyes shut, each of these strings of characters is roughly equally improbable. And improbability is another way of talking about complexity. The more improbable something is, the more complex it is. So both of these strings of characters are complex, but there's clearly a qualitative difference between the top one and the bottom one, right? The one on the bottom is complex and also functionally specified. That is to say, the arrangement of the characters is important. It's necessary for that string to perform the communication function that it performs. So in a specified complex sequence, the arrangement matters. The ordering matters. So why am I saying all this? Just to make this simple point, that when we're talking about information in biology, we're talking about information of the second kind, not the first kind. We're talking about specified arrangements. The A, C's, T's, and G's along the spine of the DNA molecule have to be arranged in a very precise way in order to generate the right sequence of amino acids. Remember, I was talking about this last night, and I have a visual aid for you. It said on the box, ages 2 to 4. Okay, so I hope you're not, well, I'm not trying to insult anyone's intelligence. It just helps me understand this. Okay, so proteins are long chain-like molecules. We saw this last night on one of my slides. They're made of smaller subunits called amino acids. And I've got these snap-lock blocks to illustrate this. There are 20 different amino acids, so at any given site, you've got to have the right one. The DNA, the information on DNA tells the manufacturing apparatus in the cell called a ribosome and some other parts which amino acid to attach at each site in a growing chain. If the amino acids line up properly, if you get the right amino acids to the right place, then the forces between them are just so to allow that chain to fold into a precise three-dimensional shape. So DNA is directing that process of protein assembly. of protein synthesis, getting the right amino acids in the right place so you get the right three-dimensional shape so the protein performs the job that's necessary. And there are thousands of proteins that we rely on for that. Thank you for the water. Yeah. Okay, so that's what DNA is doing, but here's the point. Anytime you want to build a new animal, you've got to have new information. Why? Because every new animal depends on new types of cells. So many of the complex animals that arose in the Cambrian had guts, for example. Guts require digestive enzymes. Enzymes are proteins. So if you want to have a gut, you got to have unique cell types to service that gut. Each new cell type has new proteins to service the cell type. Where do the proteins come from? The DNA, the new code. So if you want to build a new animal, you've got to have new code. That's the point. Lots of it. Every new animal requires new cell types. Every new cell types requires new proteins. Every new protein requires more genetic information. So the Cambrian explosion is not just an explosion of new animal form. It's an explosion of new biological information. Form requires information. Okay? So that's the big mystery. Just as I was talking about last night, where did that information come from? That's the big mystery. And that turns out to be a very acute difficulty for a number of reasons. Here I'm just illustrating this is one way of thinking of the metric, how much information. Sponges, which did exist in the late Precambrian, had between 6 and 10 cell types. One of these Cambrian fish, about 60 to 90. Arthropods, between 60 and 90 cell types. That's a big jump in the complexity as measured by the amount of genetic information required. Now, why is the origin of information such a problem for Darwinian evolution, or modern neo-Darwinian evolution, which emphasizes natural selection acting on random mutations? Well, mutations are copying errors in the instruction set in a section of DNA. And if you think about that just intuitively, with our own experience of computer programming, for example, if you've got a section of a computer program and you start randomly changing the zeros and ones, the digital characters, is that program more likely to, is the information in that program more likely to be degraded, or are you more likely to generate a new program or operating system from that? Now, it's obvious, right? The computer programmers are nodding, you know. Just intuitively, you're going to be degrading the information you have, okay? And there's a reason for that. In all typographic or alphabetic or combinatorial systems, if you're familiar with that term, and I'll get to that in a minute, but in all systems that are expressing information using symbols, there are a whole lot more, there are vastly more ways to go wrong than there are to go right. We know this intuitively. You start changing, you know, if you're playing Scrabble and you start changing letters, swapping them out at random, you're going to very quickly lose your triple word score, whatever you've just earned, okay? And that's because there are exponentially more ways of arranging characters that don't have any meaning than there are of arranging characters that do have meaning or will perform a communication function. That's true in a computer environment. That's true in a linguistic environment. Now, there was a conference back in 1966 called Mathematical Challenges to Neo-Darwinism, where some MIT physicists, engineers, and computer scientists challenged their biology colleagues to start to put numbers on the scenarios that they relied on to explain the origin of major complex organisms in the history of life. In other words, they were challenging the evolutionary biologists to quantify the probability and indeed plausibility of the neo-Darwinian explanation of new proteins, new genes, and new animals that depend on them. And this conference really was very interesting. You can read the proceedings today, and many of the problems that were raised there are still with us. And the mathematicians and computer scientists were convinced that if DNA is really a typographic system, if it's really digital in character, and it's conveying information and instructions, then copying errors are not going to be the way to generate new information. The odds of that, they calculated and argued at this conference, were overwhelmingly against success. Now, at that time, it wasn't possible to know exactly whether that was true or not, because there were some things we didn't know yet about DNA and proteins. In particular, we didn't know if what was true about linguistic systems and computer code is also true of the information in DNA and proteins. That is to say, we didn't know if there are vastly more ways to go wrong than there are to go right. We suspected it. The engineers and the scientists from MIT who were challenging their biology colleagues in 1966 suspected it. But they weren't sure because the experimental work hadn't been done yet to determine, essentially, how rare the functional arrangements of characters are compared to all the possible ways of arranging the characters. Okay? I have a colleague, Douglas Axe, who took this question on in starting about 1990. Some work had been done at MIT in the late 80s, early 90s, suggesting that the intuition of the mathematicians and computer scientists was correct. That functional arrangements of A's, C's, G's, and T's, that is, sections of DNA that would make a protein, were really rare compared to all the ways of arranging A's, C's, G's, and T's that would produce biological gibberish. Am I making myself clear on this? Okay. So, but we didn't know for sure. And there were some scientists at MIT who started to investigate this, and they were getting results that confirmed that intuition. Yeah, the functional arrangements are rare. It's going to be hard for random mutations to look around and find another functional arrangement. They're so incredibly rare, okay? But I have a colleague, Douglas Axe, who got fascinated with this, and he set about doing research on this at Cambridge starting in 1990. And he was asking a very important question, in fact, what I've been alluding to. How rare or common are the functional sequences among all the possible combinations of amino acids in a protein, or of all the possible ways of arranging the nucleotide bases, the A's, C's, G's, and T's in the DNA? So in other words, he was saying, for every one of these proteins that folds up into a nice structure that'll do a job, how many arrangements of amino acids are there that are just not going to fold because the force interactions between them are wrong? How many limp chains are there for ones that'll fold into functional three-dimensional structures? And he came, and this was this wasn't one experiment you could do. This was a really methodical program of research that he conducted over about 14 years. And his conclusion was this, that for a functional protein of about 150 amino acids in length, which is a modest length protein. Most proteins are on average about 300 amino acids in length. For a modest length protein, the ratio of functional folded structures to non-functional gibberish, biological gibberish, was about 1 over 10 to the 77th power. Okay? To put that in perspective, there are 10 to the 65th atoms in our galaxy. Okay, so this is a huge. Think of it as a vast haystack, bigger than our galaxy, where you're looking for one tiny little needle. All right. Now, let me maybe pump our intuitions a little bit and make this a little more accessible. You've got a bike lock, okay, with 4 dials. How many different ways of arranging the dials are there? 40, right? 10 plus 10 plus... No. Okay, I tricked you. Okay, it's 10 times 10 times 10 times 10, okay? This is what's called a combinatorial system. There's a lot of different combinations. That's what makes a combination lock so hard to crack, right? There's so many more ways to go wrong than there are to go right. Okay, it's kind of making sense now what I've been talking about? So the combination lock is kind of like the gene or the protein, all right? Because there's a lot of ways of arranging the different combinations at each site. There's 20 different amino acids at each site. So you could think of this guy as kind of like a long combination lock. In fact, I had my artist render a bike lock like this to get the point across. This is a 10-dial lock. So now you've got 10 dials with 10 digits on each, so it's 10 to the 10th power possibilities. 10 times 10 times 10 times 10 times 10, out to 10 times, okay? So here's my illustration. Imagine you're a thief out there behind the church, and clearly not a parishioner, all right? And you see that someone has locked up a very attractive racing bike, and you would very much like to have this for your own. And you know that church people are rather trusting, but there is a security guard, and he comes around about once every hour. You think, all right, 10 plus 10 plus 10 plus 10. I think I can do this, because you're a thief that doesn't know your combinatorials, right? So you start, and you're just trying them by random, and you think, well, I'm going to get there. I know I'm going to get there. But unfortunately, there's 10 to the 10th possibilities here. That's 10 billion possibilities. If you got only an hour, here's the question. Is it more likely than not or less likely than not that you're going to solve the combinatorial problem by trial and error? It's not only less likely than not, it's overwhelmingly less likely than not, okay? In other words, it's much more likely that you're going to fail than you are going to succeed. So if you're a betting person, what's the reasonable bet to make? The thief's going to make it or not? He's not going to make it, okay? Now, I'm heading someplace with this, obviously, okay? There's some principles of probabilistic reasoning, and I have learned these from our genius mathematician, Bill Dembski. He discussed this at length in his book, The Design Inference, this way of reasoning. When you're trying to decide whether or not a chance-based hypothesis is plausible, you have to assess not only how improbable the event is, but how many opportunities you have for the event to occur. And that's why I set up my thought experiment this way. It's not just that it's improbable that the thief would crack it on one try. It's even improbable when you take into account all the different opportunities the thief has to crack the code, or crack the lock. Now, obviously, we could set it up differently. We could go back to the 4-dial lock and give the thief 3 weeks. In that case, it's overwhelmingly more likely that he will succeed rather than fail. So to assess whether a chance-based hypothesis is more likely to be true or false, we need to know how improbable the event is that needs to happen, but also how many opportunities there are for the event to happen. That's called the probabilistic resources. Okay? Now the point is that in light of Doug Axe's number and other similar results, he's not the only one to get a result like this, 1 over 10 to the 77th power, it's overwhelmingly more likely than not that the mutation random selection mechanism will fail to produce even one gene or protein given the whole multi-billion year history of life on Earth. There's not enough opportunities in the whole history of life on Earth to search but a tiny fraction of that space of 10 to the 77th possible combinations that correspond to every functional combination. Why? Well, just one little number will help you put this in perspective. There have been only 10 to the 40th organisms living in the entire history of life on Earth. So if every organism, when it replicated, produced a new sequence of DNA to search that space of possibilities, you'd only search 10 to the 40th of them. 10 to the 40th over 10 to the 77 is 1 over 10 to the 37, which is 10 trillion trillion trillion. In other words, if every organism in the history of life had been searching for one of those gene sequences we need, you'd only search 10 trillion trillion trillionth of the haystack, which makes it overwhelmingly more likely, exponentially more likely than not that the mechanism will fail. And if the mechanism is more likely to fail to produce even one gene or protein in the known history of life on Earth, should we believe that that is the way that life arose? Or that the new genetic information necessary to new forms of life arose? No, we should not. Why? Because scientists want the best explanation. They want to elect the most reasonable explanation, not one which is overwhelmingly more likely to be false than true. And this brings us back to good old Jim Carrey from last night. Remember? Okay? That is if you get in a discussion about probabilities with someone on the materialist side of the argument, they will pull a Jim Carrey on you. They will say, oh, so you're saying there's a chance. But a mere finite probability does not render a hypothesis plausible. To be plausible, it has to be more likely than not to be true. And that means the mechanism that you're invoking has to be more likely than not it has to be more likely to succeed than to fail. And that's exactly the opposite in the case of the Darwinian mechanism with respect to the needed information to build even a single gene or protein. Do we get it? Okay. This is real. I mean, if you reason about this probabilistically in a rigorous way, the refutation of the mutation selection mechanism is really powerful. And I'm really indebted to Bill Dembski, who has developed these principles in a rigorous way based on his specialty in probability and statistics. And I think it's created a lights-out argument against the efficacy of the mutation selection mechanism as a means of generating biological information. Now, there are several other problems, and in my book I go into all of these. If you want to build an animal, it's not just the problem of building the information for the gene. There's higher levels of information called epigenetic information. You also have to solve the problem of getting the mutations to take place at the right time. If you want to build a new animal, you have to have mutations that occur early in development, so they affect all the events downstream as animals develop from embryo to adult. And yet those early acting body plan mutations, as they're called, are exactly the mutations that are always lethal. And so in the book, I go through a number of other problems. This combinatorial problem, the basic problem of generating genetic information is the core problem, but there are other really formidable problems for the Darwinian mechanism that are being recognized in the peer-reviewed literature. But in interest of time, I'm going to skip over those and get to the positive conclusion about design. I did mention last night that there are evolutionary biologists who are so dissatisfied with the standard neo-Darwinian mechanism that they are either calling for or attempting to generate new theories of evolution. And I list 6 of these on the screen. I have a whole chapter about these in the book as well. And I encourage you to delve into this. It's very fascinating. And many of these theories do have advantages over the old line neo-Darwinian approach. But what I show in the book is that regardless of those advantages, each of these theories, new theories, also fails to explain the origin of the genetic and higher level, called epigenetic information necessary to build a new animal form. Typically, for example, with self-organizational theory is a great example. The self-organizational theorists point to processes of self-organization that describe how animals develop. But all those processes only ensue once you have a huge complement of genetic and higher level epigenetic information. So if you can help yourself to the information that you need to build an animal, then describing how the animal gets built is no problem. You can say it self-organizes. But it only self-organizes if you already have that information. And so you need to explain where that came from. Okay? So in any case, we're back to a big question. Where does the information come from that is necessary to build these new animal forms? And I talked last night about my own story of investigating whether or not you could formulate the design hypothesis as a positive scientific argument. And I talked about how Darwin himself used this method of inference to the best explanation. We can't go back and observe trilobites coming into existence. We can't go back and observe the first cell arising. But what can we tell about its causes by looking at the effects that are left behind, the clues that are left behind? And what can we infer about what kind of cause would have been best? And I also discussed this principle that Darwin used that he got from Lyell. Darwin called it the vera causa principle. And that goes all the way back to Newton. And it's the idea that, again, if we want to explain an event in the remote past, we want to have a true cause. We want to cite a true cause, a cause which is known to produce the effects in question. Or how Lyell put it, we're looking for causes that we know from the present are sufficient or capable of producing the effect in question. We want to look for causes now in operation. And as many information scientists have noted, information always arises from an intelligent source. It always comes from, as Henry Klausler put it, from conscious and rational activity. So when we recognize that DNA is functioning like a section of computer software, and that further, when we look at something like the Cambrian explosion, we're looking at a huge infusion of new information, the argument for intelligent design is strengthened even further. Because if information always requires an intelligent source, and we can discern these huge infusions of new information at different junctures in the history of life, such as in the Cambrian period, then the best explanation, by reference to that vera causa principle, is intelligent design, because intelligence is the only known cause of huge infusions of information such as occur in the Cambrian period. So I argue that the Cambrian explosion, being an explosion of information, is another great event in the history of life which is clearly best explained by intelligent design. In my first book, Signature in the Cell, I looked at the origin of the very first life. Now we come down the timeline further and see that we've got a huge infusion of new information in the Cambrian fossil record. And I argue there that information is best explained by intelligent design. And I do so for very much the same reasons I explained last night, that applying the Darwinian method of inference to the best explanation, where the best explanation is the explanation that draws on a cause which is known to have the power to produce the effect in question, we can infer intelligent design because only intelligence, only conscious and rational activity is known to have the power to produce the key effect in question, namely the information necessary to build these animal forms. Now, someone asked me if I was going to talk about the reaction to the argument in the book, and I would like to do that in the 1 or 2 minutes we have left. One thing I didn't get to talk much about in the presentation is is the circuitry that's involved in building animal forms. This is called a developmental gene regulatory network, and this diagram, which looks like an integrated circuit, is actually from a technical paper by one of the leading developmental biologists at Caltech, Eric Davidson. And basically what Davidson and other developmental biologists have discovered is that genes are involved in integrated circuits that control how the cells that make up different animals differentiate from one another and how those cells get organized into those distinctive body plans that I've been talking about. So you have genes that are involved in networks where you have one gene and its protein product then acting to turn on or suppress another gene, and in turn causing another gene product to be produced which suppresses another part of the gene or activates another part of the gene. So you have this complex choreography of genes turning on other genes at just the right time. And that helps, that's what's going on, that's what is controlling the way animals develop from embryo to adult form. So it's this beautiful, complex choreography controlled by these integrated circuits of genes and gene products. It's just absolutely fascinating. But here's the rub. The same developmental biologists who have discovered these genes in operation have discovered that you cannot perturb them without crashing the process of animal development. And engineers know this principle. It's called the principle of constraints. The more integrated a system is, the more difficult it is to perturb it without defect to the whole. And the same thing applies to these circuits inside animals. They're exquisitely finely tuned, exquisitely integrated. And so if you, when they perform directed mutational experiments on these, invariably the circuit crashes and the animal development stops. So it's known as the problem of embryonic lethals, all right? So you got a problem. If you need one of these developmental circuits to build another, if you want to build a new animal from a pre-existing animal, you need a new developmental gene regulatory network. You need a new circuit. But we've discovered you can't change the circuits without crashing animal development, without killing the animal. So evolution at that point stops when you have dead animals, okay? So how do you ever get from one complex animal body plan to another when you can't change the circuitry which is required to build a new form from an old form? Big problem. I discuss this in the book. Okay, here's the story. Book comes out June 18. By June 19, we already have 10,000 word reviews on Amazon refuting it. Many of those were not serious. But in September, there was a very serious review published in Science. And it was published by one of the top 5 Cambrian paleontologists in the world, Charles Marshall at Berkeley, someone for whom I have a great deal of respect. Marshall's review was respectful. It was negative overall. But I appreciated it because Marshall attempted to take on the information argument of the book. And he said, Meyer claims that to build these Cambrian animals, you need a lot of new genetic information. But you don't, he says. Our current understanding is all you would need to do would be to rewire one of the genetic regulatory circuits that controls how the other genes are expressed, which build the parts for the Cambrian animals. And I read that, and I thought, yes, this is perfect. Marshall has tried to refute the argument on point, and you do not need a PhD in biology to understand that this very good scientist actually begged the question. That is to say, he did not answer the argument. A genetic regulatory circuit is made of what? Genes. Genes contain what? Genetic information. Okay? So he presupposes the existence of a genetic regulatory circuit, for one, and then says, well, if the evolutionary process could rewire it, which is first of all, a teleological metaphor, a design metaphor, but second, let's let that pass. Rewiring a genetic regulatory circuit would require coordinated mutations in the different genes involved in the regulatory network, which is to say, changing the code, which is to say, adding new information. Second source of unexplained but yet important information in the Marshall scenario. Thirdly, this genetic regulatory network acts upon other genes for building various parts of animals, something that Marshall explicitly has acknowledged in his own technical work separate from the review. So you have three sources of unexplained, significant sources of unexplained genetic information that are presupposed in this short little statement. No, our current understanding is that all we would need to do is rewire the genetic regulatory circuits so that they would act differently on the pre-existing genetic information. So in an attempt to refute the argument from information, the argument I make about the Cambrian information explosion, Marshall actually presupposes prior sources of information showing what? That our current understanding, that is in evolutionary theory, does not provide an explanation for the origin of the information necessary to build life.